Hello, I welcome you all to today's session where we are going to discuss first the continuation of the last chapter that we had discussed in the previous session. In the previous session, we have discussed about the inheritance of human skin color as an example of polygenic inheritance. So today, first we will start our discussion by comparing the monogenic and the polygenic inheritances. Before we discuss about this, you first remember that in case of monogenic inheritance, the examples will be like the inheritance of that height of the stem in case of pea plant, uh, like this flowers, axial violet flowers crossed with terminal white flowers. These all will be example. In case of polygenic inheritance, this three pairs of dominant genes controlling the expression of a single phenotype, that is inheritance of the skin color in humans, will be the example. So you remember this and let us compare. So the first point under monogenic inheritance, we should say that it deals with the qualitative traits such as the form, the color or the structure or the height, like this. Height is a confusing concept because height in case of pea plants, that is controlled by a single pair of gene. In case of humans, it is under the polygenic inheritance. So let us not discuss that as an example over here because it might confuse us further. Let us take clear cut, you can say, characters. Like in case of monogenic inheritance, you consider about the inheritance of color of the flower in case of pea plant. At one extreme, you will be finding the violet color pea plant as the expression of the dominant phenotype. At the other extreme, you will get the expression of the white colored flower as the expression of the recessive phenotype. There will be no intermediate colors or intermediate expressions like the colors of the mauve colored flowers or the lilac colored flowers or the purple colored flowers. So those things you will not find in case of monogenic inheritance. So they are examples of discontinuous variation, means there are no intermediate or gradual phenotypes, only two phenotypes, one at the dominant extreme, one at the recessive extreme. But in case of polygenic inheritance, remember that I have told you that several varieties of skin colors are possible, starting from one extreme, that is showing the expression of the pure black phenotype, and at the other extreme the pure white phenotype, and you will be getting a gradation of skin colors in case of polygenic inheritance. So having said this, the first point of difference, let us say that in case of monogenic inheritance, it is always the inheritance of quality. So it is a qualitative inheritance. In case of polygenic inheritance, it will always be a quantitative inheritance. So polygenic will always be concerned with quantity, and monogenic will always be concerned with you can say quality. So this one, mono means quality, poly means quantity. So this, I think I have made it clear. Second point, as I have written over here, monogenic traits are generally controlled by one pair of alleles, like capital T small t or capital T or small t will be sufficient for expression of the phenotype. But in case of polygenic inheritance, always it will concern about the expression of the character which are controlled by generally more than two pairs of alleles. Like in case of human skin color, we have seen it is the three allelic pairs controlling the expression of the single phenotype. In case of kernel color in wheat, you will find there are two pairs of genes which control the phenotypic expression of a single type. The third point of difference will be in case of monogenic inheritance, it will show you discontinuous variation. By this I mean that there will be two extreme phenotypes, one dominant, one recessive, and there will be no intermediate phenotype. But in case of polygenic inheritance, there will be gradual development of the phenotypic trait. So you will get the intermediate phenotypes which will lead to continuous variation. Let us come to the next point of difference between these two. So in case of monogenic inheritance, obviously there are no intermediate phenotypes. And here you will get a variety of intermediate phenotypes. Point 5 as the point of difference will be a single dominant gene is able to express itself fully. Means the single character like color of the flower, position of the flower, color of the seed, shape of the pod, that are sufficient to be expressed by a single pair of gene. So one dominant gene pair can express the trait fully. But here, as you can see, capital A can express only 33% of the trait. If you get capital B added up, it will be around 66%. If you add capital C, then only 100% phenotypes will be expressed. So here the effect is added up by the presence of various allelic pairs. The next point of difference will be there is no addition effect. Like if you get capital T, T1, T2, if these three genes will be present, the characters will not add up. So here no additive effect of the gene. But here, as I told you, presence of every one pair will add up the character further. So this will, the quantitative one will show you that is why it is called as metric or quantitative traits that we have discussed in our previous session. So here the F1, more importantly, now let us come to the cross part. So here the F1 individuals will show you parental phenotypes. By this I mean that if you cross capital V with capital V small v, all the F1 plants will be hybrid capital V small v, but they will be purple in color or violet in color as their parents are. But in case of this, it will show, the F1 generation will show you a new or intermediate phenotype. Like when we had crossed or tried to find out the outcome of a marriage between a Negroid a male and a pure white female, then in F1 generation we had got all the children who had mulatto skin color. So mulatto skin color was not present in any one of the parents. So it is a new trait. In the next point, F2 individuals, they will resemble either the dominant or the recessive phenotype. So the ratio will be clear cut. Like in monohybrid cross, it will be 3:1. In dihybrid cross, it will be 9:3:3:1. But here in polygenic inheritance, you will find that it will be like in case of human skin color, it is a trihybrid cross. So there you will find a variety of ratios possible, like we had done that 1:6:15:64 ratio, like that. So here the F2 ratio is somewhat clear cut. Clear cut, by this I mean that all the organisms or individuals or offsprings, they will resemble either of the two parents, either dominant or recessive. But here no such proposition will be there. You will be finding so many individuals who will show you a skin color that was originally not present in their parents. Now let us compare the next one. The phenotypic expression of the genotype is not affected by the environment. Like if you put more fertilizer to this pea plant, the violet flower will not become white or vice versa. So here you will find that environment doesn't have much effect on the expression of the phenotype. But here, like I had told you, that if you are fair skinned, then you can get tanned, and if you are with dark skin, then you can get lighter. So here environment plays a major role in the expression of the phenotype. Now let us come to the last point. Last, because these are the points that we have discussed already. So the progeny can be analyzed by the counts and ratios, and here the progeny can be analyzed by statistical method. So here this is a little bit tricky point for you. Here the ratio can be only found out by mathematical means, but here the ratios are more clear cut. So having said this, this brings us to the end of the series of discussions that we were carrying out on the monogenic and polygenic inheritance. Now let us come to the next topic, that is the back cross and the test cross. So exactly why we carry out this back crosses and the test crosses. The major, you can say, requirement is that, like, I will give you a violet flower plant. By looking at it, can you tell that which one is capital V genotype containing violet flower bearing plant, and which one is the capital V small v genotype bearing violet flower plant? Because both of them will appear exactly alike. So sometimes because now onwards we will be discussing about the topics like pedigree analysis, like inheritance of chromosomal and Mendelian disorders in humans, particularly about the inheritance of various diseases that we are going to discuss. Then these genotypes and phenotypes will matter to you a lot. So for that you have to have, particularly to solve the pedigree analysis type of problems, because that is one of the major spotting type of practicals scheduled for this year also. So you must be having an idea that once you are confused about the genotype of a particular organism, how you are supposed to know beyond a reasonable doubt that whether it is the homozygous dominant phenotype or the heterozygous dominant phenotype. So first, let me explain you the test cross. Once you are becoming familiar with the concept, back cross will be easily understood by you. And we will be discussing it for the monohybrid one as well as for the dihybrid one. So test cross, essentially the definition tells you that it is the cross between the F1 hybrid and the homozygous recessive parent. So generally it is a cross that you can see that ethically we cannot practice it in case of humans. So that is why, because once we go for pedigree analysis, you will find that certain genetic disorders like alkaptonuria, phenylketonuria, muscular dystrophy, sickle cell anemia, color blindness, hemophilia, they are present in the family. They run among the family tree. But essentially you cannot, like, you want to predict that if in a family the parental generations were having the problems or the expression of the phenotype, then whether the child will be affected or not. But basically in case of humans, you cannot get the child marry with the mother to see the outcome. So please understand that test cross, theoretically you can work it out in case of humans, but for ethical reasons we cannot. Having said this, the definition says that it is the cross between the F1 hybrid and the homozygous recessive parent. So now let us try to work out the cross. So here the parents, suppose it is a violet flowered pea plant. It is crossed with a white flowered pea plant. I am working out the normal cross. So the genotype of this type of parents, that will be capital V for this plant and small v for this plant. Then the gametes for F1 generation will be during meiosis, when this plant will produce the pollen grains, 50% of the pollen grains will receive this complement of capital V, other 50% will receive the other complement of capital V. So basically all the pollen grains will be having the same genotype, that is capital V. When this plant will be producing the ovule, then 50% of the ovule will be getting or receiving this complement of small v, and the rest complement of small v will be passing on to other 50% of the ovules. So the F1 generation of these two pea plants, whether this fertilizes this ovule or this pollen grain fertilizes this ovule, or this pollen grain fertilizes this ovule, or this pollen grain fertilizes this ovule. In all the cases, it will be capital V, small v for the F1 generation. So all the F1 generation, they will express what phenotype? They will be having the violet colored flowers. Suppose I have given you 10 seeds, out of which 5 seeds are having this genotype which is expressing the violet phenotype, and other 5 are having this genotype. They are also expressing the violet phenotype. So how do you know that whether it is this kind of genes that are expressing the violet phenotype, or it is this combination of alleles that are expressing the violet phenotype? Now if you want to carry the test cross, then the definition says that it is the cross between the F1 hybrid. So my parent for a test cross will be this F1 hybrid, that is a violet flowered pea plant, but this violet flowered pea plant will be heterozygous for the allele. It will be crossed with the homozygous recessive parent, so obviously this, so it will be crossed with a white flowered pea plant which is homozygous for the trait. I hope this is visible to all of you. Now if I say their genotype, so here you cannot have much permutation and combination. Definitely this I have to write over here, as this is the F1 hybrid and this is the double recessive parent. So it will be crossed with this small v, small v. Sorry, I should not have put this cross over here because already I have put it. Then gametes. So because we are carrying out test cross, we are not supposed to write whether it is F1 or F2. So this will separate from this complement during meiosis. This will separate from this complement during meiosis. So gametes, 50% of the pollen grains will be receiving this capital V. Other 50% will be receiving this small v. But all the ovules will be of the same type, because they will be 50% will be receiving the small v allele and other 50% will be receiving the small v allele, because here we are getting two different type of genotypes of the gametes. So let us have a Punnett square drawn. So the progeny generation, as we are getting two different type of gametic genotypes, so we will make a Punnett square that is having 2 rows and 2 columns. So this will represent the pollen grain genotype horizontally, and this will represent the ovular genotype vertically downwards. So here once again you see this is the genotype of the pollen grains and this is the genotype of the ovule. So here I will write capital V and small v. Remember to encircle when you are writing the gametic genotype. So it will be small v for the ovules. Now let us see what happens. If this pollen grain fertilizes this ovule, it is capital V, small v. So the expression of the phenotypic trait will be violet. If this pollen grain fertilizes this ovule, it will be small v. Expression of the phenotype will be white. If this fertilizes, this pollen grain fertilizes this ovule, it is capital V small v. Again the expression is violet. This one is also same, that is capital V, sorry, small v expressing the white phenotype. So what will be my phenotypic ratio? I am getting out of this Punnett square, I am getting two phenotypes. One is violet and the other one is white. So I am getting two individuals with this violet expression, two individuals with the white expression. So the ratio is 1:1, isn't it? So for a test cross, like if I am giving you a pea plant, seed of a pea plant, and I am letting you know that you try to find out whether it is homozygous or a heterozygous one. Then you plant the seed, let the plant grow out of it. Then you take the seed of that, or the particularly the seed of the white flowered plant, and you also grow pea plants out of it. Carry out artificial hybridization between the plant which has grown from the seeds which I have given you and the plant which have grown from the seeds of the white plant that you have collected. Carry the artificial hybridization and wait till the flowers develop in that artificially hybridized plants. So if the flowers are appearing in the ratio of 1:1, like 50% of plants are having the violet colored flowers and 50% of plants are having the white colored flowers, that means the seeds which I had given you, they were having capital V small v genotype, means they were heterozygous for the trait. And let us see the concept of the back cross. Then you will understand that if I had given you the capital V phenotype, genotype, then what would have happened. So the definition of back cross tells that it is the cross between the F1 hybrid and any one of the parents. So as for test cross, we have taken the homozygous recessive parent as the other individual. Then for the back cross, let us take the dominant one. So already, let me write it for the back cross only. So parents for the back cross will be obviously the homozygous dominant F1 hybrid. That is, for this example, if I am continuing, then it will be a pea plant with violet flowers, because all my F1 hybrid are capital V small v and they express the phenotypic expression of violet colored flowers. That will be crossed with homozygous, because it is a back cross. Let me cross it with homozygous dominant parent. The homozygous dominant parent, definitely it is a pea plant with violet flowers. So let us come to their genotype. So because it is, sorry, I should have written it heterozygous, really sorry. It should be heterozygous, sorry. So it is capital V small v and it is homozygous, means capital V. So what will be, sorry, please remember that I should have written that all the F1 hybrids are heterozygous for the trait. By mistake I had written homozygous. So here the gametes, this capital V will separate from this small v, this capital V will separate from this capital V. So 50% of pollen grains will receive this gene, 50% of the rest pollen grains will receive the recessive allele. Here all the ovules will be of one type because the plant is homozygous for the trait. So the progeny generation let us draw a Punnett square, and the Punnett square definitely will be having 2 rows and 2 columns. On horizontal side we will write the composition of the pollen grains, and on the vertical downward side we will write the composition of the ovules. So if this pollen grain fertilizes this ovule, then the plant is a violet expression flower containing plant. This fertilizes this, it will be violet, but it will be heterozygous for the trait. If this fertilizes this, again it will be the expression of the violet colored flower, but it is homozygous for the trait. If this pollen grain fertilizes this, then it is capital V small v. It will be heterozygous genetically, but phenotypic expression will be violet. So in a back cross, you are actually not getting any ratio. All the plants are showing the dominant phenotype. So now I think I have made the concept of the back cross and the test cross clear to you. If I have given you the seed of a plant that is having capital V small v genotype and phenotypically it is expressing the violet colored flower, then if you carry out a test cross, then all the F1 hybrids or the progeny hybrids, they will show you 1:1 ratio for the trait for which you are carrying out the test cross. And if you are given the seeds which are having homozygous genotype for the trait, like it is having the expression of the violet colored flower, but the genotype is capital V, then if you carry out back cross, then all the genotypes and the genotypes will show you the homozygous and heterozygous variations, but phenotypes will be the same. So this is all about the monohybrid test cross. Now let us work out a dihybrid test cross. The definition will remain the same, that it will be the cross that will be carried out between the F1 hybrid and the homozygous recessive parent, but it will be for the dihybrid cross. So dihybrid test cross. Let me carry out the simple one. So because I am directly writing the test cross, so parents will be the F1 hybrid. So let us take the famous example, that is tall plant with the violet flowers. So it will be a tall plant with violet flowers. It will be crossed with the homozygous recessive parent. So it will be a dwarf pea plant with white flowers, recessive for both the traits, that is height of the stem and color of the flower. Then the genotype, because this one is the F1 hybrid, so it will be heterozygous for the trait. For tall, it will be having the expression of capital T small t, and for violet it will be capital V small v, because it is heterozygous for the trait. I am talking about the F1 hybrid, and this one will be small t small v small v. So gametes for the expression of the progeny, this one will separate and move to 50% of pollen grains and will take this one. This one will take this one. Again this one will take this one. It is becoming more confused. Let us have this fork line method so that we don't get confused. So this takes up this 25% of pollen grains will be capital T capital V, 25 of the next set of pollen grains capital T small v, next 25% small t capital V, next 25% or the last 25% small t small v. So you will be getting 4 different type of pollen grains. For this one, because it is a homozygous condition, this T will take up this V, this T will take up this V. So all the ovules will be of capital T small v, small t small v type. So let us construct a Punnett square to know the progeny generation. So because all the ovules are having the same genotype, so let me modify the Punnett square where we will be having only one row and 4 columns. Why I am doing this? Just you follow me, you will get the logic. Here I am supposed to write the genotype of the pollen grains. So genotype of the pollen grains capital T capital V, this one, followed by capital T small v, then small t capital V and small t small v. here the ovular composition is only one, na? So that is only the small t, small v. So if this fertilizes this, it is capital T, small t, capital V, small v. So capital T will dominate over the small t, and capital V will dominate over the small v. So the phenotypic expression will be tall and violet. If this pollen grain fertilizes this, it is capital T, small t, expressing the phenotype of tall, and small v, when they are coming together, they will express the phenotypic expression of white. Now if this pollen grain fertilizes this ovule, then small t will be coming together, expressing the phenotypic expression of dwarf, and capital V, small v coming together will express the violet expression. If this pollen grain fertilizes this, it is small t coming together, again expressing the dwarf phenotype, and small v coming together, expressing the phenotypic expression of white. So don't you feel the ratio over here is 1 is to 1 is to 1 is to 1? So dihybrid test cross will show you the ratio of 1 is to 1 is to 1 is to 1. If I will carry out the back cross, then all the genotypes and phenotypes, genotypes will be showing you that dominant and recessive expressions, but the phenotypes will show you the same expression. So having said this, once again let me recapitulate what I have discussed so far. I have told that when I have given you a particular trait and you know the phenotypic expression but you are not sure about the genotype, then do carry out the test cross to know beyond reasonable doubt that the trait which I have given you, the phenotypic expression, whether it is due to the presence of homozygous or same type of alleles, or presence of heterozygous or different type of alleles. Because please remember, like in the examples we have discussed, capital T, small t and capital T will express the same phenotype, but genotypically they are different. Similarly, capital V and capital V, small v will express the same phenotype, but genetically they are different. Having said this, I think I have been able to make you properly understand the concept of test cross and back cross. Thank you.